and today we're going to be talking about using the Cancer Genome Atlas as a tool to improve undergraduate student understanding of cancer genetics and the hallmarks of cancer progression. So let's go ahead and get started. So ultimately, this study surrounded the idea that undergraduate students, like most, if not all of us in this journal club have common misconceptions about important cancer hallmarks. Oftentimes, this is because the understanding of cancer outside the field of oncology is very limited. So in fact, cancer has had its definition as a disease that was caused by abnormal cell growth for a pretty long time. However, we've only recently started to see and appreciate the idea that cancer is a group of diseases that is characterized by rapid cell growth and division, as well as a group of diseases that are able to spread to different organs in the body. So let's go ahead and talk about some background on common cancer hallmarks. So these complexities, as I mentioned previously, were more significantly started in the 1980s and 1990s to be recognized. Here we truly began to understand how cancer cells metastasized. Such studies gave rise to a very important aspect of modern cancer diagnostic processes, Hanahan and Weinberg's seminal review of the hallmarks of cancer. This review that they did detailed 6 properties of cancer cells, and in 2011, it was later expanded in 2011 to contain 8 vital hallmarks for recognizing the presence of cancer. So what are the hallmarks? These include sustaining proliferative signaling, evading growth suppressors, resisting cell death, enabling replicative immortality, including angiogenesis, activating invasion and metastasis, deregulating cellular energetics, and avoiding immune destruction. So what exactly are we talking about in terms of gauging how we can expand people's knowledge about the hallmarks of cancer, and more specifically, undergraduate students who are in relevant fields to cancer research? So student-centered learning is becoming a more popular method for engaging with the material in a direct manner. In a sense, hands-on learning. So how this hands-on learning was carried out in this study was through a tool called the Cancer Genome Atlas, also known as TCGA. And I will expand more on this tool in subsequent slides. So who exactly participated in this study? So to begin, participants were 2nd and 3rd year undergraduates in a genetics and cell biology course. This course was done in an all-male college and included both a lecture and a laboratory section. It's also to be noted that these people were involved in both fall 2019 and the 2020 semester. So we're going to dive into physical methods of how the study was conducted. Before then, does anyone have any questions that I can answer? If not, we can go ahead and proceed. Okay. So let's go ahead and start with the physical methods. So looking at the structure of the course, as mentioned previously, students participated in this study and they were enrolled in a specific biology course. This course ensured that among both semesters of the study, so fall 2019 and fall 2020, there was equal emphasis on lecture content and study materials. One of these cohorts, however, participated in an additional bioinformatics-based project, which will be the focus of today's methodological analysis. As the slide indicates, the project analyzed TCGA data and was used to learn about the genetic influences on cancer hallmarks. So when we think about it, one cohort did have a little bit of this extra enrichment that was also part of the study. This is important for later results. This project involved numerous tools for gene analysis, one of which being the CBio portal for cancer genomics. This tool was used to investigate mutations and copy alterations in tumors. Another tool being used was GEPIA, This tool studied RNA sequencing data in order to identify the differences in gene expression between tumors and normal tissue. It is also to be noted that students were provided with future optimal use of these tools for accurate results. So when looking at what was being analyzed by the students, it was said that each student selected 3 cancer types that had available TCGA data for analysis. I would like to note that these students are part of the cohort that had that extra enrichment laboratory. So for each cancer type they selected, students were asked to investigate genomic alterations by identifying the 5 most common mutations and the 5 most common copy number alterations. Students were also asked to investigate the differences in protein expression between normal tissue and tumors for each cancer type chosen. This included 5 upregulated genes and 5 downregulated genes to identify alterations in its copying mechanisms, which is basically transcription, that were identified with cancer. Search engines PubMed and Google Scholar were also used to research functions of these 20 genes and determined which hallmarks are likely to be influenced by the alterations and transcription alterations. If you have been keeping tally, the 20 genes stems from combining the 5 most common mutations, 5 most common copy number alterations, the 5 upregulated genes, and the 5 downregulated genes. So as we know, this study is not necessarily focusing on the novel discoveries of the student from the project, but whether their understanding of cancer hallmarks improved by doing the project and the course. So a pre and post assessment of the student's knowledge on cancer hallmarks and the genetic base of cancer development was conducted on both the cohort with the project and the cohort without the project. This assessment contained multiple choice, true-false questions, and short answer questions that required students to describe the 8 hallmarks of cancer. So we're going to talk about results next. Before that, if you have any questions about the methodology of this presentation, you go ahead and unmute and ask any questions. If not, we can go ahead and proceed. Okay, so let's go ahead and talk about results. So what exactly did this study conclude? So in regards to the pre-assessment, it was discovered that there were, in fact, misconceptions students had in regards to the complexities of cancer and the genetics behind cancer. As shown on the slide, a major misconception regarding genetic factors in cancer development was identified. For 40% of students surveyed in the pre-assessment believed that gene mutation was the only genetic factor in cancer development. Furthermore, in the pre-assessment, about 46.7% of students surveyed were able to correctly identify cell proliferation and evasion of cell death as hallmarks of cancer. However, overall, at the start of the semester, only 26.7% of students surveyed could correctly identify 2 hallmarks. I would like to note that 15 students were surveyed and involved in this project. Since the average class size in the college was about 14 to 18 people, it just happens to be that the class at that time was about 15 people per semester. So what happened after the project and semester? What did we learn? So as shown by the slides, much improvement on both an individual and group level were demonstrated. In the post-assessment, about 86.7% of students correctly identified all 8 hallmarks of cancer. No student correctly identifying less than 6 hallmarks. On overall question accuracy, there was also noted improvement, for accuracy increased from 39.3% accuracy to 75.6% accuracy. In addition, 26.7% of respondents had originally stated that differing genetic abnormalities were responsible for cancers, so no single mutation is responsible. This number increased to 66.7% following the post-assessment. So let's talk about this figure here. So kind of looking at this graph here, we can see the drastic increase between the pre-project assessment and the post-project assessment in both graphs. So in all of these graphs, there's a large difference in the pre-project and post-project assessment percent of correctness and number of correctly identified hallmarks. So in graph A, you can see how the pre-project assessment, at most, students were getting 2 hallmarks correct. This most likely correlates with the cell proliferation and invasion of cell death. Those were the most common ones students were able to identify correctly. Versus afterwards, in the post-project assessment in figure A, where we see that the average student identified all 8 hallmarks. Of course, there were some abnormalities with students choosing 7 or 6, but no student got fewer than 6, showing a marked improvement. This is also demonstrated in graph B with percent correct, and graph C also has a similar structure, showing the increased percentage that students were being accurate in their assessment of hallmarks. So if we're looking at this figure here, these figures actually show the difference in the lecture only and lecture and project cohorts. So as I mentioned earlier, one cohort only had the lecture component of the study versus another cohort had the lecture and the project, which is the TCGA analysis. So as we can see, this figure details the idea that there were noted differences in those who only received the lecture instruction and those who only participated in the project. We can see that those who participated in the project identified approximately 7.8 hallmarks with a noted percent error of 0.14. Those receiving only lectures correctly identified 4.13 hallmarks correctly. So their p-value, which is basically a statistical significance value, is less than 0.001. This makes the relationship statistically significant and shows how there's a marked relationship in between hands-on learning and improvements in understanding of material. Project participants also scored higher on the multiple choice and true-false portion of the assessment than those in lecture. So therefore, we can clearly tell that the access to active engagement and observation of principles regarding cancel hallmarks is extremely influential to the learning of said material. So these are just some sample responses of students from the pre and post assessment. I believe this was the extended response portion, where they asked students to essentially describe a hallmark of cancer. So in the pre-assessment, there were some that were identifying the hallmarks of cancer. So for example, student number 2 correctly identified that the evasion of cell death, which is apoptosis in this case, was a hallmark of cancer. So the ability to avoid apoptosis is a hallmark of cancer. However, noted by their post-project assessment, they were more able to thoroughly describe their hallmark and the science behind that hallmark and what exactly it means versus just identifying it. So we can truly see that the project assessment, so going through this lecture and going through maybe this project, maybe they were part of the project, really helped to solidify that understanding. So going beyond the study, there are several factors we can kind of talk about. So how can we kind of engage these results in improving the education of cancer hallmarks? So we can incorporate active and student-centered approaches to cancer education. And we can also focus on finding the magnitude of misconceptions students hold. So there are some biological principles required for the transition from a healthy single cell to metastatic disease that were not correctly identified. And we can correctly identify those misconceptions and focus on those throughout our education. There's also an array of genetic anomalies that drive hallmarks to cancer development, and students were not able to initially identify this. And so being able to find these misconceptions and target those in education is extremely important. We can also go through and identify the common characteristics shared by diverse cancer types. So focusing on the themes. So for example, a TP53 gene was frequently mutated among different types of cancers in the TCGA data that was analyzed. Obviously, not all of the cells analyzed had this, but seeing that this common theme arose in this study will help students to be able to identify the fact that maybe there's certain biomarkers and genes in their coded proteins that will be useful for future developments, such as effective cancer therapy. And finally, just in general, making learning more interpersonal via some freedoms. So, for example, students had a choice in the cancer studied and the methods used, which could be a great way to stem interest and keep the students engaged by being able to apply material to things that they already are familiar with. So those are all factors that go beyond the study that will be extremely useful. So with these results in mind, I do want to make some remarks and maybe potentially open the audience for this as well. So there are some ethical considerations we do have to kind of identify in this study. So two principles I thought about when analyzing the study were the importance of considering confounding variables and the potential for research bias. So, confounding variables are essentially variables within an experiment that a researcher fails to account for. This is often seen as the third variable, not explicitly recognized, and it varies depending on the context of the experiment. So, for example, the study was conducted in a small men's-only college. So, even though there may not be a specific correlation, we do have to think about, is there a component gender plays in retention of material? Obviously, we may not be different neurologically, but do things such as gender bias and gender stereotype affect the performance of participants? Or even if we were to put this study in another college, would there be a difference in male and female retention of material, maybe due to gender bias and gender stereotype going on within the college, if there is any present? Another example would be within the present data being analyzed. It was assumed that the TCGA data being studied was completely correct. However, if hypothetically there were errors within the data, how could that affect the study? So are these databases that students are using completely correct? So that also could be a confounding variable that could vary the experimental results. It is also important to note that oftentimes studies may not even remotely hint at a confounding variable. For example, the time of day that the performance assessments were given could have affected the experimental results, basically their scores. So personally, I'm not a morning person, so taking the exam early may have affected my score if I were a part of this. So it should also be considered that maybe the time of day at which these performance assessments were given could actually have affected how these students performed on the each of these assessments. It was also not explicitly stated whether or not the same time was given in the pre and post assessment. So if the students took one in the morning and one in the afternoon, that could have varied the results. As I previously stated, it's not explicitly mentioned, but it is something to be considered when we're doing further studies. So we can also think about this in a more general sense and possibly even apply it to the medical field. You know, we're all part of Caduceus Club here. So there is not a single pathway to diagnosis, just like there isn't a single path to cancer development. So therefore, we truly have to consider all possible variables when conducting research and medical work. Obviously, this is most of the time impossible to do all factors. However, when repeating experiments and going through and reanalyzing data, confounding variables are something that must be paid attention to. Another factor that I wanted to highlight here is the research bias part of the experiment. So the article indicated that the study allowed for students to pick their 3 different cancers, which many and most of them were working with because they were familiar with someone they knew they had it. So basically, these cancer cells that people were working with, the students were familiar with it just because they had a family member or a friend who was diagnosed with this type of cancer. So like on the slides, being able to incorporate personal experiences with interest is a great way to build passion and connection to research. However, this may not be what we want as a researcher. So personal interest, for example, if the person has a decent understanding of the cell biology and cell principles behind that specific cancer type, may cause an unwanted jump to conclusions without truly making these discoveries. This goes hand in hand with the fact that ultimately research bias towards certain predicted outcomes can take away from the novel study. So good data is phenomenal, obviously, and sometimes students may tend to take data that they are familiar with and want to make conclusions off of that just because they know that their conclusion is more likely to be correct because they're familiar with it. But more often in the scientific field, no matter what field you're going into in terms of research or clinical diagnosis, data is messy and will need to be repeated for accurate results. So it's crucial to factor in your own discovery, for such leads to the best productivity.
If you don't mind me interjecting for a second.
Yeah, sure.
So regarding the confounding variables, I agree that they probably should have done a more broad research across both genders and perhaps more countries around or more colleges around the country. But again, like it might have been like a local research without the resources to do that. I also want to mention that another confounding variable could be each student's course load, since that might have varied in difficulty, which could have affected that experiment for sure. And on top of that, I also have a question regarding the pre and post project assessments. So I remember reading in the article that the assessments were actually voluntary. And I just want to verify that did all of the students respond to it despite the fact that it was voluntary?
Yeah, great question. So actually, I am not too sure as to whether or not all these students responded. I don't think that was explicitly stated when I was going through there. But those are some great questions. And I do thank you for interjecting on your opinion of those confounding variables, and that is very important to consider too, that like you mentioned, there are just so many factors and we are not able to necessarily limit them all and account for them all, but such things such as coursework, or maybe there was the resources were limited and that's why the study was done like that. Those are all great variables that could affect experimental outcomes. So I do want to thank you for taking the time to recognize that. Those are some great opinions. Does anyone else have any thoughts to share? Okay, well, I am going to go ahead and stop sharing. That's kind of all I had, but if we want to talk about this more, the article, I believe, is available. I believe Carl was able to share that out. And yeah, I cannot really see the screen right now, so if there's any questions in the chat, maybe I can go ahead and answer those as well. I know not all the environments not necessarily people to share, because I know sometimes it gets finicky. So if anyone else does not have any questions, that's kind of all I had for everyone today. So thank you for listening. I do appreciate you guys coming to Journal Club and sharing this time with me to look at research and look at confounding variables, research bias, and just continue to broaden our knowledge on these topics in general. Anything else?
Nothing for me. Thanks for the presentation and good job on that.
Yeah, no problem. Thank you.
If you don't have anything else, I'm going to end the meeting in like a few seconds here. So we're all good.
Thank you.
All right, great.